All right, so urinary system. Okay, so first thing is anatomy. And I'm not going to spend too much time on some of the gross anatomy because that is part of the lab. So I'm going to let you review the lab, and with it, just review these gross anatomy organs. Now, functions of the kidney. Let's see. The kidneys filter blood. The filtrate, the substances collected from blood, are going to be modified in the kidney. The good stuff will be put back into circulation, back to the blood. The bad stuff is going to be excreted as urine. When all is said and done, out of all of the fluid filtered from blood, only 1% of that will be secreted as urine, which means that 99% is going to be put back into the blood. So this is one of the main jobs of the kidneys, but that's not the only job of the kidneys. Because the kidneys are excreting urine, and urine is mostly water, and that water came from the blood, the kidneys are essentially regulating blood volume. In regulating blood volume, they regulate blood pressure. So that is one of the most important jobs of the kidney. Another function of the kidneys is to regulate blood solutes. Again, because the kidneys are filtering blood, anything that is in the blood, that means ions like sodium, calcium, potassium, chlorine, bicarbonate ions, glucose, which I know is not an ion, but nutrients, glucose, bilirubin, anything that's in the blood is regulated by the kidneys. So if the blood needs or the body needs more sodium, then it's going to excrete less sodium in the urine, for example. Another job of the kidneys is to regulate pH of not only says extracellular fluid, it also means blood. It also regulates blood pH. And it does that by regulating hydrogen ion concentration, which is what pH is. So keep in mind the jobs of the kidneys. To top it all off, the kidneys also have an endocrine function. They secrete renin, which is a hormone that we talked about, that is involved in regulating blood pressure. It's part of the renin-angiotensin mechanism. Also erythropoietin that we talked about, or EPO, that stimulates red blood cell production. It's also involved in activating vitamin D, which eventually becomes a hormone. So those are the endocrine functions of the kidneys. Again, I'm going to let you review the anatomy. And again, the anatomy of where the kidneys are located, their retroperitoneal organs. One of the things about the kidneys is that they're very, very well protected. They are perched on the walls, on the posterior walls of the abdominal cavity. And they're surrounded by fat tissue and connective tissue. So that if you were to do a dissection on the abdominal cavity of a human or an animal, and you were to remove all of the organs inside the cavity, you still would not see the kidneys because they're completely covered with fat tissue and connective tissue that have to be peeled away to finally find the kidneys. So they're very, very well protected organs. Yeah, we talked about what the renal ptosis is when one of the kidneys falls, or because they are perched on the wall of the cavity, and they're held by connective tissue and fat. Sometimes maybe the person experiences a sudden weight loss. The kidneys may fall from their location. That's called renal ptosis. Hydronephrosis is when the kidneys begin to accumulate fluids that may be due to an inflammation or due to some type of blockage. And hydronephrosis could lead to necrosis or death of the kidneys tissues. Now, the internal anatomy of the kidneys. When you cut a kidney, you will see on the periphery of the kidney, outermost layer, a light tissue, which we call the renal cortex. And then deep to the renal cortex, there's the renal medulla, which consists of a series of pyramid-shaped structures. So this is a nice specimen here, and this is a figure of a kidney. One thing about the kidneys, again, very well protected. This outer layer here is the renal cortex, which is a layer of connective tissue. This lighter layer here, I'm sorry, not the, sorry, that's not the capsule. I'm looking at something else. Give me just a second to do this here better. This is the capsule. That's the renal capsule, which again is connective tissue. The cortex will be this lighter layer right here. All through that is the cortex. Okay, you get the idea. That's the renal cortex. And the medulla, where the pyramids are, right there you can see the pyramids. In between the pyramids, the lighter areas are called the columns. All right, and then you can see in this picture, at the tip of the pyramid, there's this cup-like structure, which is going to collect urine and take it to larger areas of collection, which in turn takes it to this funnel-shaped structure called the renal pelvis that is going to drain the urine into the ureter. The tip of the pyramid is called the papilla. So, again, the outside is the renal capsule, the cortex right here. The renal medulla is deep to the cortex, and medulla consists of pyramids and columns. The papilla, the renal papilla, is the tip of the pyramid where we have a cup-like structure that collects the urine. That cup structure is called the minor calyx, and these minor calyces drain into a major calyx, which in turn drains into the renal papilla, which is this funnel-like structure right there. Okay, so you should know the anatomy of kidneys. Pyelitis, pyelo, means renal pelvis. So this will be an inflammation of the renal pelvis. Pyelonephritis, on the other hand, is a lot more serious. Pyelo is renal pelvis. Nephritis refers to the nephrons, which are located in the cortex and pyramids. So this is referring to an inflammation of cortex, pyramids, and renal pelvis, which is essentially the whole kidney. Now we're going to go into the microscopic histology of the nephron and of the kidneys. And the microscopic anatomy of the kidneys is made of these units called nephrons that function to filter blood and modify the blood into urine. So the nephron is the histological and functional unit of the kidneys. This is the working horse of the kidneys. Each nephron consists of a renal corpuscle, which is found in the cortex, a proximal convoluted tubule, which is found in the cortex, a loop of Henle, which is found in the pyramids, and a distal convoluted tubule, which is found in the cortex. So this is more or less what a nephron looks like. The blood that the nephron is going to filter comes in via one arteriole called the afferent arteriole, which brings the blood into this capillary structure called the glomerulus. Blood exits the glomerulus via the efferent arteriole. So the renal corpuscle is made of the glomerulus plus the Bowman's capsule, which is this cup-like structure right here that collects the filtrate. So the glomerulus is where filtration takes place, where blood is being filtered and blood components are being pushed out into the Bowman's capsule. The Bowman's capsule collects filtrate and begins to circulate it in the tubules, in the renal tubules, starting with the proximal convoluted tubule, which leads to the loop of Henle, and then which goes into the pyramid, comes back from the pyramid, goes into the distal convoluted tubule, and then finally drains into a collecting duct, which is not part of the nephron. So there are millions of nephrons per kidney. They are small, about 55 millimeters in length. There are two types of nephrons, juxtamedullary nephrons and cortical nephrons. In humans, most of our nephrons in our kidneys are cortical nephrons, 85% versus 15% juxtamedullary nephrons. And this is the difference between them. A cortical nephron is located in the cortex and fully in the cortex. It has a short loop of Henle into the pyramid and then comes back to the cortex. A juxtamedullary nephron, the nephron is located closer to the medulla. That's why it's called juxta, meaning close to, medullary. It has a very long loop of Henle which goes deep into the medulla, into the pyramid, sorry, and then comes back from it into the distal convoluted tubule, back into the cortex. So that's the difference between those two. You should know the difference. This is another PowerPoint here that shows you cortical versus juxtamedullary nephron. And I like this picture because it is showing you yet another difference between them. Notice that around the convoluted tubules, the proximal and distal, the cortical nephron, which is the most abundant nephron in the human kidney, has these capillaries wrapped around called peritubular capillaries. These capillaries will be collecting the parts of the filtrate that need to go back to the blood. And the juxtamedullary nephrons are going to have a long loop of Henle, and the capillaries are not going to be around the proximal or distal convoluted tubules. They're going to instead follow the loop of Henle in a parallel fashion. These capillaries are called the vasa recta. Again, that's just another way to look at the nephron. Let me point out a couple of things about the nephron, which are actually very, very important. Number one, notice that the afferent arteriole is larger than the efferent arteriole. And that is important because what that means is that blood that goes into the glomerulus is not going to come out of the glomerulus easily, which means there's going to be a backup of blood in the glomerulus. It's almost as having a double door going into a room and a tiny little exit door. People, you fill the room with people, and when it comes time to exit, the number of people that exit is going to be less than the number of people that go in, creating a very full backup in the room. There will be a lot of people, and just I imagine these people running around, bumping into walls, trying to get out, bumping into each other, creating pressure. So that's what that backup of blood is creating inside the glomerulus, is increasing blood pressure, which is going to cause filtration, the pushing of things out into the collecting Bowman's capsule. Okay, so that is not fortuitous or random. The fact that the afferent arteriole is bigger than the efferent arteriole, it is purposeful. The other thing to note is that the distal convoluted tubule, as it comes back from the loop of Henle right here, it is positioned strategically in between the two arterioles. And that again is not fortuitous. There's a reason for that. This right here is a control center, which is going to control blood pressure. It's going to control how the kidneys are functioning. Okay, so it's going to control renal function. All right, so this is a close-up look at the Bowman's capsule. And notice that the tissue that makes up the capsule is simple squamous epithelial tissue. The tissue that makes up the glomerulus is a capillary. All capillaries are made of simple squamous epithelial tissue. You can see the afferent arteriole much bigger than the efferent arteriole. This is a cut right here through the distal convoluted tubule as it travels in between the two arterioles. And notice all the cells in the area. Notice, for example, that the cells in the distal convoluted tubule are different on one side and the other. On one side, they're big square cells. On the other, they become smaller little cells. These smaller little cells are macula densa cells, which are reading the concentration of the filtrate as it travels through the distal convoluted tubule. And that will be a measure of the blood pressure in the glomerulus. Juxtamedullary cells, again, are monitoring the conditions of the filtrate and the conditions of the blood as it goes into the glomerulus. This entire area, which is a control center, is called the juxtaglomerular apparatus. And this is, again, the control center for the local control center for the nephron. So this is what is going to adjust filtration to accommodate blood pressure, changes in blood pressure, for example. And we'll talk more about that, if not today, then next time. So this is the glomerulus right here, which is a fenestrated capillary with fairly large pores that allows for the exit of substances from the blood. However, the glomerulus is completely covered by these little cells called podocytes, which have extensions of their cytoplasm called pericells. And these extensions intercalate into each other, creating filtration slits, so that only things that are small can be filtered. So it goes the size of the substances that can be filtered. So large substances like albumin should not be filtered out of the blood. So again, that's showing you the filtration slits right there. So the filtration membrane, similar to when we're talking about the respiratory membrane, anytime you have diffusion or filtration, this movement of molecules back and forth, you need a very thin membrane. So the same thing is happening with the filtration slit. You have simple squamous epithelium of the Bowman's capsule. You have simple squamous epithelium of the endothelial cells of the capillary, of the glomerulus. There are little pores in between cells. Those are the fenestra. These are fenestrated capillaries. And then the filtration slits of the podocytes further provide a sieve through which large molecules cannot pass. So obviously cells are not going to be filtered out. Albumin is too large. It should not be filtered out. So any large protein should not be filtered out. We may find a little bit of it in the urine, but not very much. So again, that's just another look at the juxtaglomerular apparatus. Okay. Which again has the macula densa cells, the juxtamedullary cells. Let's see. I don't quite see them here. I'm sure they're in there somewhere. All right. The arterioles, in this case, the afferent arteriole is down here, also have cells that control blood or that sense baroreceptors that sense blood pressure. Those will be the granular cells. All right, so that's just more of the same. These are the filtration slips made by the podocytes. Okay, so in the Bowman's capsule is going to catch the filtrate that is being filtered from the glomerulus and is going to deliver that filtrate to the proximal convoluted tubule. So this is the PCT or proximal convoluted tubule. And the cells in the PCT are going to be simple cuboidal cells. Simple cuboidal cells. The example for simple cuboidal cells last semester were kidney tubules. So those are the cells of the PCT and DCT. So these are square cells that have lots of microvilli for increasing surface area. So these are cells that are going to reabsorb the substances that have been filtered out and need to go back into the blood. Substances like water, glucose, vitamins, amino acids, some of the ions like sodium, all that will cross these simple cuboidal cells on their way back to the blood. And that's called reabsorption. Notice that these cells are close together and they're glued together so that things cannot go in between cells. And again, the purpose of the microvilli is to increase surface area. And then we're going to the descending loop of Henle. In the descending loop of Henle is going to have segments of simple cuboidal epithelium and very thin segments of simple squamous epithelial tissue. I'll come back to this in just a second. And then we go into, from the loop of Henle, we'll go back to the distal convoluted tubule. So this will be DCT. And in this case, again, we have cuboidal cells, but these cuboidal cells are not going to have microvilli like the PCT cells. They are going to have a lot of mitochondria, which means that whatever movement of substances back and forth are going to be due to active transport instead of just reabsorption by diffusion, for example. Okay. So the difference between PCT and DCT, this is a slide very much like the ones we saw last semester of simple cuboidal epithelial tissue. So this is a DCT tube, and you know it's a DCT tube because there are no microvilli in the cells. This is a PCT tube, and you can see have a sense of the microvilli in there. So again, PCT versus DCT. Our collecting duct cells are very similar to the DCT, and there's going to be no microvilli, and basically just cells are going to transport based on a active transport. So you can see here actively reabsorbed. Again, the process is called reabsorption. Ions like sodium, potassium, and chloride. The renal arteries deliver about 1/4 of the cardiac output to the kidneys each minute. So the kidneys are receiving tremendous amounts of blood. We're going to see the arterial flow and the venous flow into and out of the kidneys, and they're going to be very similar. The nerve supply of the kidneys is via sympathetic fibers to form what's called the renal plexus. So the kidneys is mostly under sympathetic control. Collecting ducts are going to collect the filtrate from many nephrons. So these are not part of the nephron. And the collecting ducts end into the papilla that is going to deliver the urine, that's going to be called urine, into the minor calyces. In the collecting duct, we have two populations of cells. The principal cells are cuboidal cells without microvilli that are involved in maintaining water and salt balance. So these cells are going to be sensitive to hormones like aldosterone and ADH. On the other hand, intercalated cells are cuboidal cells with microvilli that are going to involve in the maintenance of pH. So there's going to be the secretion and reabsorption of bicarbonate ions and hydrogen ions. So principal cells control water and sodium, potassium via the actions of ADH hormones and aldosterone hormones. So these are cells that are sensitive to hormones, whereas the intercalated cells maintain pH by reabsorbing and secreting bicarbonate ions and hydrogen ions. The cells of the distal convoluted tubule are very similar to the collecting duct cells. So again, that's just an overview of the kinds of cells in the nephron. And then when it comes to blood vessels of the kidneys, this is exactly the same as what you guys should have done in the lab. So just go back and follow the circulation of the kidneys from renal artery to segmental artery to interlobar artery to arcuate, cortical radiate, and then from the cortical radiate, just try that again. From the cortical radiate will branch the afferent arterioles into the nephrons. So the cortical radiates are like a little tree, with the little branches being the afferent arterioles. Then coming back via peritubular capillaries into cortical radiate veins, arcuate veins. There are no interlobar veins, but directly into the interlobar veins. There are no segmental veins, but it instead goes directly into the renal vein. So definitely know this flow of blood. Notice from afferent arteriole to glomerulus to efferent arteriole to peritubular capillaries, and from there into the venous circulation. Okay. So this is a view of cortical radiate artery. Arcuate artery here. Cortical radiate branching out, and from it branches the afferent arteriole, which goes into the glomerulus, exits through the efferent arteriole, and from there is going to go to the peritubular capillaries. Okay. We talked about these. The glomerulus is a capillary that is specialized for filtration. And filtration means pushing by blood pressure. And that's why we need a high blood pressure in the glomerulus so that filtration can take place. Part of what creates that high blood pressure is the fact that the afferent arteriole is larger than the efferent arteriole. What is the difference between renal pyramids and medulla? The renal pyramids are in the medulla. So medulla is the area where the pyramids and the columns are located. Okay, yeah. So, because the efferent arteriole is so small, there we go. Blood pressure is low because there's low blood volume. Remember, high blood volume, high blood pressure, low blood volume, low blood pressure. So there's low blood pressure because there's low blood volume. That means that the pressure in the peritubular capillaries is also very low. So the peritubular capillaries are low blood pressure vessels, which is exactly what we need if those capillaries are going to absorb some of the nutrients that were filtered out. So they're going to cling to the renal tubules, these peritubular capillaries. So here's the peritubular capillaries clinging to the tubules, and blood circulates very slowly through those capillaries, allowing time for reabsorption, collection of substances that shouldn't have been filtered but were filtered back into the blood. Okay. Yeah, the high resistance in afferent and efferent arteriole causes blood pressure in the efferent arteriole to decline. All right. We talked about the filtration membrane, which allows the passage of small things. However, proteins in blood should not be filtered. They're too large. Not to mention things like cells should not be filtered either. All right, so in this chapter, terms are important. So when we talk about the filtrate, we're talking about the substances that are being pushed out of the blood and collected in the Bowman's capsule, which is essentially a lot of water. It's blood plasma minus the proteins. So it's going to be the plasma, water, no albumin, no fibrinogen, no clotting factors. However, yes, glucose, yes, vitamins, yes, ions, amino acids. So all this filtrate has to be processed, and only less than 1% of it is going to be excreted as urine. So most of it is going to have to be reabsorbed, 99% back into the blood. And that's the job of the tubules, kidney tubules. So urine is formed by filtration first, then in the tubules, there's reabsorption going on. Also in the tubules, there's secretion going on. And then finally, the excretion of the urine by the urethra. So these are the processes that are taking place in the nephron. Glomerular filtration takes place in the between the glomerulus and the glomerular capsule or Bowman's capsule. So notice that this is the only place where filtration occurs. Then the filtrate begins to travel to the tubules. And in the tubules, reabsorption will take place, which is the returning of the substances that are important back into the blood, and secretion, which means some of the substances that should have been filtered and were not, let's say substances that are waste products that might have been too large to be filtered, are going to be moved by active transport from the blood into the tubule, and that is called secretion. So you need to keep these terms straight. This is probably the most important aspects of the chapters, to keep the terms straight. remember what is reabsorption, which is number 2, versus secretion, which is number 3. Okay, and students get those two confused all the time, and you shouldn't. Those are the important terms. Okay. Let's see. All right, so glomerular filtration is a hydrostatic pressure event, which takes place due to blood pressure, and it's the pushing of substances out into the Bowman's capsule. Glomerulus is a filter essentially, and only small things can be filtered. The hydrostatic pressure that allows for this filtration is about 55 millimeters of mercury. Actually, it's not 55 millimeters of mercury, but the pressure in the capillaries is higher than the pressure in other capillaries. Is 55 millimeters of mercury instead of about 30 millimeters of mercury, which is the blood pressure in most capillaries. So the glomerulus has a higher blood pressure, which can account, that can do the job of these filtration. All right, the actual final pressure that is going to push things out is only 10 millimeters of mercury. So let me go ahead and go over here. And essentially what will be happening is, again, because the afferent arteriole is bigger than the efferent arteriole, blood accumulates in the glomerulus. And you increase blood volume, you increase blood pressure. And so the blood pressure in the glomerulus will be increased from about 30 millimeters of mercury in normally, so this will be here coming in 30 millimeters of mercury, to 55 millimeters of mercury. So that is this pressure, this is the force that is pushing things out. However, for every force that is a counterforce. The counterforce is the colloid osmotic pressure, which is due to albumin, keeping things from being pushed out. So the force of albumin is 30 millimeters of mercury. So that's the counterforce. So the 55 millimeters of mercury in one direction are offset by 30 millimeters of mercury in the opposite direction. So that leaves us with 25 millimeters of mercury of force pushing out, except there is, as fluid collects in the Bowman's capsule, it also exerts a pressure. Those molecules are pushing against the glomerulus and are returning some of these substances back into the glomerulus. That force is 15 millimeters of mercury. So that leaves us with 10 net millimeters of mercury pushing things out. So what that means is this difference in pressure is actually very small, and any change in blood pressure will affect the functioning of the kidneys. So you can change blood pressure systemically by just 10%, so that instead of being 30 millimeters of mercury, it's going to be 33 millimeters of mercury, which again will make the blood pressure in the glomerulus 10% higher, bringing it up to 60 millimeters of mercury, which now we have 35 minus 15. It will give us about 20 millimeters of mercury at the end, which would be too high and it could damage the filtration membrane. On the other hand, if blood pressure goes too low, so let's say instead of 55, it goes down by 5 millimeters of mercury to 50, the albumin doesn't change, so that one stays the same. Capsular hydrostatic pressure will be about the same. It may go down a little bit also, but let's say it's about the same. So now you're left with 5 millimeters of mercury of net filtration pressure, which is not enough for the kidneys to work. So for the kidneys to work, they have to keep the pressure in the glomerulus at 55 millimeters of mercury. Any deviation from that pressure would compromise the workings of the kidneys. So the kidneys are very sensitive to changes in blood pressure. The glomerular filtration rate is the volume of filtrate formed by the kidneys per minute, which is about 120 to 125 milliliters per minute. What determines the GFR is the filtration membrane and the filtration membrane permeability. Obviously, if the GFR goes up, the net filtration pressure goes up. If the GFR goes down, the net filtration pressure goes down. In this case, more pressure is not good because it will damage the filtration membrane. Filtration membrane is very thin. If the GFR is not high enough, then the net filtration pressure will be too low for filtration to take place. All right, so glomerulonephritis is a damage to the glomerulus, which in this case, if the glomerulus is damaged, now the filtration slits will be damaged and large substances will be filtered out, and we'll be able to see those large substances in the urine. Okay. So the glomerular filtration rate is tightly controlled by the kidneys themselves, because that's how the kidneys keep themselves functioning. So this is the main control right here, day in and day out. This is the main thing that is happening to the kidneys, is controlling its own pressure. So as long as blood pressure is maintained within a normal range, between 80 and 90 to 180 millimeters of mercury, the kidneys can function fine, because they can auto-regulate. The two mechanisms of regulation, myogenic mechanisms and juxtaglomerular apparatus mechanisms, or GGA. And this is the way it works. It's actually quite clever. If systemic blood pressure increases, that is dangerous for the kidneys, because that could damage the glomerulus. High blood pressure could damage glomerulus. GFR will increase, and substances will be filtered that shouldn't be filtered. In order to prevent this from happening, the kidneys constrict afferent arterioles. That constriction is going to lower the blood going into the glomerulus, and if you lower blood volume, you lower blood pressure. And that's going to bring the pressure in the glomerulus back down to a normal range. On the other hand, if blood pressure is too low, now filtration cannot take place. The kidneys will shut down. So the response of the kidneys is to dilate the afferent arteriole. That is going to increase blood volume going into the nephron, which increases blood pressure. So that's what the kidneys do to control blood pressure and continue functioning. The macula densa cells read the flow of the filtrate inside the distal convoluted tubule. And if the flow rate becomes too high, indicating high blood pressure, the GGA cells release a vasoconstrictor that is going to make the afferent arteriole become smaller and lower the blood volume, with that lowering the blood pressure. So there's the macula densa cells that control this mechanism. So this is local control. And as I mentioned before, local intrinsic or local control is what prevails most of the time under normal circumstances. Extrinsic control occurs when something is happening systemically. We have a systemic problem. And unfortunately, or that's just the way it is, not fortunately or unfortunately, when something is happening that is systemic to the entire body, the regulatory mechanisms of the body are going to prioritize the important organs. The important organs are going to be the heart, the brain, the lungs, and the skeletal muscle, because now we're under sympathetic control, flight or fight. The kidneys are not a priority. So extrinsic control in response to a systemic problem is going to affect kidney function. The sympathetic nervous system is no friend of the kidneys. The kidneys are not important under flight or fight. So this is what the sympathetic nervous system is going to do. Okay. Yeah, so this is what we're saying before. Under normal conditions, autoregulation prevails. All right. Under stress, flight or fight, sympathetic stimulation, norepinephrine is released. Epinephrine and norepinephrine are released as hormones. Both are going to cause constriction of the afferent arteriole, and that is going to lower the blood pressure in the glomerulus and stop filtration. That's going to trigger the release of renin by the granular cells of the juxtaglomerular apparatus. Renin is going to initiate a cascade of events geared to increase systemic blood pressure. So when we're under stress, kidneys are going to be shut down, and that's going to cause the secretion of renin, which will be geared to increase blood pressure. So that's just, again, a reminder of where the juxtamedullary apparatus was located. Okay, so remember, under stress, the granular cells are going to secrete renin because the blood pressure is low. And now this renin will put in place a mechanism that will be geared to increasing blood pressure. This is what we call the renin-angiotensin mechanism, which we already talked about. The renin-angiotensin mechanism is stimulated, and let's see, it's going to lead to wider reabsorption in the kidneys in order to increase systemic blood pressure. Okay, so let me go back and clarify, or at least talk about something important on these. Stress. What's stress for the body? Low blood pressure caused by hemorrhage, caused by shock, infection. That's going to stimulate sympathetic nervous system. The sympathetic nervous system will shut down the kidneys by constricting afferent arterioles, because the kidneys are part of the peripheral nervous organs, peripheral circulation, and that's going to cause the kidneys to secrete renin. Renin converts angiotensinogen in the blood to angiotensin 1. Angiotensin 1 circulates in the blood and in the lungs due to the actions of angiotensin converting enzyme will be converted to angiotensin 1. Angiotensin 1 will be converted to angiotensin 2. Okay. Angiotensin 2 is a hormone that is going to increase blood pressure because it is going to activate secretion of ADH by the posterior pituitary, activate secretion of aldosterone by the renal cortex, increase thirst, more vasoconstriction of peripheral blood vessels, which means, this right here means afferent arteriole is going to be constricted even more. creating even further shutdown of the kidneys. So when we say that the renal angiotensin mechanism increases blood pressure, it's going to increase blood pressure for everywhere else in the body except the non-important organs: kidneys, skin, GI tract, reproductive organs. Those organs will still have even lower pressure. So stress, low blood pressure, shock can kill the kidneys, because now blood pressure will fall even more in the kidneys in an attempt to bring blood pressure up everywhere else in the important organs, or lowering blood pressure even further in other organs. Nonetheless, this is the extrinsic or systemic control, because in this case, the body is trying to keep itself alive, and the kidneys are not in the list of crucial organs. Okay, so I'm going to leave it here. We have another PowerPoint to go, and we can tackle that one next week. And we'll have this chapter finished next week, and we can start reproduction, and then finish reproduction on the last week of class. Redo this schedule. We only have a couple weeks left, so I'm going to redo it so that we can fit in these material in this last weeks of class. Any questions? Okay, I'm going to go ahead and exit, and I will